Describe the steps of protein synthesis, beginning with the attachment of a messenger RNA molecule to the small subunit of a ribosome and ending with the release of the polypeptide from the ribosome, including your answer a discussion of how the different types of RNA function in this process. Translation can be broken down into three different stages: initiation, elongation, and termination. Let us begin with initiation. In the question, the stage of initiation has already been reached. The stage begins with the attachment of the mRNA molecule onto a small subunit of the ribosome. The actual synthesis itself is brought on by the start codon AUG, being met by an initiating tRNA complementary to that codon. This tRNA molecule enters directly onto the P site of the ribosome. The first amino acid is always methionine. Furthermore, initiation factors play an important role in the first stage of translation. These factors are a set of proteins that facilitate the events that occur in initiation. The ribosome itself is divided into three compartments: the A site, the P site, and the E site. With initiation complete, now let's move on to the second stage of translation: elongation. As you might have already predicted from its name, the elongation process is the synthesis of the polypeptide chain itself. This will happen as the tRNA constantly brings in different amino acids into the ribosome. However, the elongation process is not as simple as that. There are many smaller bits and pieces that make up the process of elongation, such as the elongation factors, for example. These factors are a set of proteins that orchestrates the events of the elongation process and ensures 99.99% of the accuracy of the translation. While the peptidotransferase is an enzyme which forms peptide bonds between the adjacent amino acids in the growing polypeptide chain. With the formation of the peptide bonds, the tRNA exits through the E site. Now we have reached the third and final stage of translation, called termination. Termination is the stopping of all translating activity due to one of the three stop codons entering the A site of the ribosome. The stop codons are UGA, UAA, and UAG. There are no corresponding anticodons for stop codons. With termination, the polypeptide chain is released and the mRNA breaks down. Then the tRNA is freed to the cytoplasm of the spore. tRNA has such a significant role in translation. However, we are yet to discuss what tRNA actually is. Well, tRNA stands for transformed nucleic acid, and they are the transporters of free-flowing amino acids in the cytoplasm to the ribosomes for synthesis. And unlike mRNA, which breaks down tRNA is recycled and used over a number of translations. So then what is mRNA? Well, mRNA stands for messenger ribonucleic acid, and it is the platform in which our proteins base themselves on. Having been transcribed from our DNA in a stage preceding translation or transcription, it carries our genetic information. Now for the third and final type of RNA, or rRNA. rRNA stands for ribosomal ribonucleic acid, and they're such an essential part of translation because protein and rRNA makes up our protein factories, ribosomes.